amazing introduction. It's funny, I wouldn't have considered myself a nerd, but then you read all that out and it's like, I am a nerd. And I'm proudly nerdy, actually. It's also been one of those long days, so I'm not really sure if I can multitask a microphone and a clicker, but I'll give it a go. So as Nicole said, that I have been involved in this kind of work for 15 years, and I gave it this title, The War of the Worlds, because I'm sure all of you have seen the movie when you've got the giant aliens coming from outer space, and the last one I think I saw was Tom Cruise, and I know that he's weird, but you know, they devastate everything. These aliens from outer space, big, huge things. No one understands how they can take them down. They end up being taken down by a tiny microscopic organism. And that's kind of what's happening in most of our native forests at the moment. They're really under attack by these invading, except it's the other way around. It's these tiny things coming in and invading our forest and getting a hold and spreading around there and really taking down some of our iconic taonga species, because we call them taonga species, so they're treasures. And once they're here, there's really not a lot we can do about it. So that's why I've titled this, and the reason I've been working in this area, I kind of came from a different area, but I really had an SOS from my good friend and colleague working at the Auckland Council. He knew my skill set, he said, Kauri, look, come here, I know what you can do, can you help us out? We're desperate. So I answered his call, for better or worse, and this is kind of a little bit about my journey going through this, and hopefully it's not funny, like it's really hard to make something like this funny, but you can laugh at my expense. I've got some jokes in there at my expense, so you can do that. But I'm kind of hoping that you'll just get a little sense of what's involved in the work and maybe get curious about it, I suppose. So just introducing our unique kauri forest. And just by the way, has anyone actually been up to Northland and seen some of those kauri? Beautiful, impressive, aren't they? Massive. Yeah, and so that's always awe-inspiring when you go there and you just actually take it all in, soak in these big giants, and the big ones that we know of have names too. They've got separate names that the iwi up there give them. But weirdly, naturally, and we don't know why this is, that yellow line that you'll see, and I won't use the laser pointer because that's, again, one too many tasks for me today, but above that yellow line, that's where they naturally exist now in New Zealand. Then the thing about these big trees is that they are really long lineage, and when I say lineage, that they actually go back to the Jurassic and the Cretaceous, so they covered most of the globe, and right now what we see is left are these kind of remnant fragments in the southern hemisphere. And there's about 22 of these big species. And so New Zealand kauri has relatives in New Caledonia, it has Australia, it has Fiji, and it also has, if you're familiar with the monkey puzzle tree, that kind of crazy looking tree that's massive and big. So if that's from Chile and Argentina, that's a native there. And they're all related, and they're part of this ancient lineage that goes right back to the Jurassic. And so here they are now kind of sitting in pockets around the world in these little remnant forests, whatever's left of them. But one of the cool things I liked about trying to help these iconic forests is to get a sense of the time, because yes, they're an old lineage, so they've been around since the dinosaurs, and if you look at them you can kind of tell that these things would have been with the dinosaurs because they've got tough leaves, and so something that would have eaten them had to be tough and big themselves. But also the fact that they're so old lived as well. I mean, we don't even know how old some of these are. They're probably like 2,000 to 4,000 years, but we're not sure. And the size of them, I think the biggest one we have currently has around 16 metres of girth and 30 metres of height. So you can imagine the kind of carbon that's stored in these trees and all the things that hang off them and live too. So they create their own special ecosystem because they're so long-lived. They hang around at the landscape and they drive everything around them. So that's what we call their keystone species. And some of them have been there, would have seen the Haast eagle, they would have seen more. And when you stand there and you're sampling around them, because I'm interested in soils, that's what I do, you know that at some point a moa or a Haast eagle would have flown over, and that's kind of cool. And that kind of relationship is what's talked about in a lot of the iwi there. They believe them sentinel beings, and that they're kind of almost as static as, say, the mountains themselves. But there's just some of the cool pictures of the plants that co-evolved with these trees as well. So when we do see a lot of these trees disappearing in other forests due to removal or due to kauri dieback, a lot of these plants go with them, and so it irreversibly changes the landscape. Unfortunately, it makes it a little bit more weedy. like I kind of touched on, and parihaka is a very different kaupapa from this one, so we're not talking about that one. But these trees, because they're so old, they have stories around them, what we call pūrākau, and they don't make sense to people, and I know scientists have tried to pick this apart, but the honest truth is that's how a lot of Indigenous communities make sense of the environment around them. And the reason why I have a picture of a southern right whale is because in Māori mythology, that is the brother of the kauri tree. And the reason why they're brothers, and actually if you go around the Pacific, and I do work in the Pacific, a lot of Pacific communities have the same kind of stories, that every plant species on the land has a marine equivalent. And you'll see that in Hawaii, and you'll see it in Samoa and Fiji as well. But the legend goes that the two actually were on land and they walked side by side. But the whale wanted to go back into the sea. And the kauri itself said, look, brother, I think you're cold. So how about, sorry, it's the other way around. I'm getting confused. I told you it's been a long day. So the kauri wanted to stay on the land, and the whale's like, brother, I think you're going to get cold, so here, I gift you my skin. And if you look at the bark of the kauri, it's very scale-like, much like a fish itself. And so that's where the legend comes from. And that's why some of our normal practitioners, for lack of control or solutions they have for kauri diet, have gone to look at some of their rongoa, which uses whale, and they add that with other bioactives, and they've been applying that to the tree. So some of that you'll see here. And for them, it's a way of actually contributing and helping to the ecosystems because they haven't seen anything else done. And so that talks to a little bit about Māori acting as kaitiaki in this space, but also leaning on that pūrākau that talks about the whale as well. Feel free to jump in and ask me questions. But the reason why we've got to this point where we have kauri dieback is an issue, isn't a new one in terms of our forest, and it's not a new narrative either. Now, I've got pictures of houses there, which is streets in San Francisco, right? And I've got forest below it. So prior to about the 1800s, a lot of our landscapes look like this. Those were kauri trees that lined the Hokianga, and that's probably in 1880, I think. So that's what it looked like, right? And the kauri probably covered about 1 million hectares of forest. So now we're really looking at less than 0.5% of its natural coverage. And that map next to you kind of shows you what's left. And some of those purple bits are really the only bits we have left. We have the big trees as well. And those are the areas that I've worked in. But what happened is that we came over, we cleared the land, we started putting down farms, we sold a lot of the logs overseas, and that went to fit out the British Navy, but also San Francisco. And that in the earthquake, San Francisco was devastated. I think it was in the 1930s. Got my times wrong. But we sent most of our forests over there to rebuild San Francisco. So that's why I have the pictures of that. And also, just out of interest, that the Ranfurly Shield was made out of kauri as well. So a lot of our, and I wish the government wouldn't do this, but it keeps going on selling our stuff overseas. So it keeps going to take everything that we can't replenish and send it overseas. So I really hope they've got something better up their sleeves other than that for economic growth. Sorry, I wouldn't get political, but I did. Just because I see it happen over and over again, it's the answers like you're just taking stuff and there's not much left to grow back. Anyway, so this is what happened. In the 1950s, a bunch of hunters, hunt pigs, they were on Aotea, which is Great Barrier, and they noticed some of our big kauri trees were bleeding out. They had these lesions around the base, and they didn't know what it was, and at the time, I think they would have sent something to DSIR, which was completely different to anyway. Anyway, that's where plant people were based and scientists, and we didn't have molecular techniques back then, and so it was misclassified as something completely different. And they misclassified it as it was called a Phytophthora heviae. Now, I know I didn't have the pit bull, pitta bull. Like, I can't even brain that today. But it's difficult enough with the Phytophoras. And this is what the organism is that came over, and it's likely in the 1950s, and I'll talk to a little bit about that. But that's what came over. And for a long time, up until 2015, it was thought it was Phytophthora heviae. And I don't know if you know anything about Phytophoras, but it is the organism, and it's a oomycete, and it's similar to fungi, but it's not fungi. But fungi are actually more closely related to us, and I know some of you are thinking, yeah, I can tell. But it's something completely different. And just also out of interest, if there's any geneticsy people in the audience, the size of the genome of a kauri tree is about 30 billion base pairs, right? And the human genome is 3 billion base pairs. So you can get an idea of how hard it is when someone says, oh, let's just sequence it. I mean, that's a lot of effort to do that. And another cool, interesting fact is that we share 50% of our DNA with plants. And again, I think you're probably thinking, I can tell that with some people, right? I know when I told someone that, they were thinking, I actually know someone like that. So that's harsh. But coming back to the molecular story, is that we only had the kind of diagnostics to pick it up in about the 2000s, when we started developing these things called PCRs and primers and things like that we're able to take out pieces of DNA that we could actually recognise that belong to this species. And so, long story short, it wasn't Hevea. It was a brand new Phytophthora to science, and it was called Phytophthora agathidicida, and my colleague sequenced it and gave it that name. So what that actually means, it's a Greek word, and it's like plant destroyer, and agathidicida is the scientific name for kauri, and so that's what it's called. And actually the Phytophthoras are what took out the potatoes, so that was partly responsible for the Irish famine. I think the politics were the other part of it. So that's our interest. That's how difficult these organisms are. Yeah. They're also really, really tricky to do something about. That other diagram, and I promise I didn't have too much sciencey stuff in here because it's night time and we're drinking, except me. But it has 6 life cycle stages. And so when you do develop tools to target these pathogens, each one of these tools has to target a certain life cycle. So what do you do? And when you have no money and no time, it's you kind of just got to put your money into some sort of direction. And I think, good, the video has showed up. And so that little video, and this comes to War of the Worlds, has tiny, tiny zoospores. I don't know if you can see them moving, but that's what infects kauri. These tiny, tiny, less than your hair wide, that's what's sniffing out the roots. And they sniff out the sugars which are secreted through the roots. And any damage to roots, that's what they're looking for. And once they're in there, like that diagram on the other side says, they encyst the roots and they just grow up into the tree and they starve it. And so often when you see trees starting to thin out at the crown and kind of look a bit dead, like that poor specimen in the corner there, that's what happens. They starve them from the top down, and that's called crown death. So that's what you're seeing. And once they're in a tree, there's nothing you can do about that. I know someone talked about phosphite. Yep, they do that, use that in avocados, but I mean, you're talking about trees that have 16 metres girth. I mean, if you put in phosphite injections there, I think you'd kill the tree just injecting something in there, actually getting in the first place. So this is the kauri dieback, and it was officially diagnosed in 2015, and ever since then we've kind of been in some sort of crisis management mode. And that's my colleague who convinced me to come on board, Nick Waipara there. He works at the Auckland Council. I don't know if I thank him or be mad with him. But anyway, we've been on this journey together for a long time. So you're wondering, what are the current management strategies? And the truth is, not much. We have people and these very fancy walk stations, mostly in the Waitakere ranges, and it has this trigene disinfectant in it where you kind of scrub your boots and spray something. And I know I was talking to the cameraman who's doing his masters in compliance in this area, only New Zealanders are the worst at this. They're the worst people at taking care of their own stuff. The tourists are good at it, but the locals are the worst. But this is all we've got, this trigene that actually we don't even know if it's going to kill the little spores, the survival spores that hang around in the soil. So that's part of the problem. And so the only other tool that's been brought out is called the rahui, which is the ban. And that, I was involved in a lot of the Waitakere Rangers management crisis meetings. And so what the iwi did there, which is Te Kauramaki, they put a ban or rahui on a lot of these tracks. And I know you said you had trophies for your email weird insults. Man, we got them too. Yeah, we got them, the anti-vaxxers, the anti-climate change. So we called them the kauri dieback deniers. That's what they were. We were denying their rights to enter the forest. And I got CC'd on the mayor's emails, the minister's emails. It was hilarious. But they're out there. They're out there. I didn't keep a trophy of them. I just blocked them. Sorry. And so that's all there is, apart from surveillance. And that's all that's happening. And there was a point where there was a flourish of funding to do something, but it's kind of all dried up. And what happens is when you get a flourish of funding, you get all sorts of scientists putting up their hands thinking they have the answers, and some do, but also some don't. And so they spread it so thin that nothing happened. And this is kind of where we've left it. It's called a cliff of finance or funding stream. So it's depressing talk, and it's not really funny, but it's true. And I kind of don't know how to talk around this apart from this is what's happening. So what could I do as a frustrated scientist with some skills? And so what I did is I got together with other colleagues, and one of them is a geneticist himself, and the other one works at Ancient DNA down in Otago. And what we got together is trying to develop a diagnostic kit, because at the present, the only way you could detect Phytophthora agathidicida, or kauri diabet we like to call it, was through this really laborious plant baiting exercise which took days and hundreds of dollars. And who in the community would have hundreds of dollars per soil sample to try and figure out where this is? And also, you're covering thousands of hectares, right? So we decided to produce a lateral flow system, which is using a CRISPR-based technology, and it is very specific. So if it was in the soil, we were going to find it. And we are nearly there. And it's so community. Thank you. Oh, is that you, Georgie? Yeah. Oh, haven't seen you in years. So we wanted to put this into the hands of the community so at least they could do something about their own situation and try and manage it themselves. And that's kind of our contribution to this all. But there's another side that we've been developing as well, and this is trying to look at biocontrol tools. Now that's a really sort of interesting space, because this was also led by the iwi, trying to help us understand what natural tools were in the environment that we could work with and deploy. And this is another sort of friend of mine. Basically, we were sampling the soils around the kauri trees, and the iwi knew that there were some really healthy soil spots and some not so healthy. And there was spots where there was kauri diabet and not. We didn't know why, and this is literally needle in a haystack stuff. But what we managed to do is we cultured out a couple of cool organisms. One's a penicillin, surprise, surprise, and the other one was another bacteria. It's called Burkholderia. But anyway, this is needle in a haystack stuff. And so what we did is we found that it kind of worked against Phytophthora agathidicida. But you can't really use that, and you can't culture that out, because long story short, if you add something to soil, it's like a drop in the ocean, right? And so that's not really a tool you can use, but it gives us some insight into some other bioactives that we can use. And of course, when you say you've got a problem, people think that there's lots of money to be made, and so you get a lot of snake oil people. And I know, sorry if I offend anyone here who's using snake oil, but we have a lot of people, can you use geranium? How about thyme essence oil? And you know what? I had a master's student who tested all this, and of course it doesn't work. But what does work is we asked stuff you can't get in New Zealand, but we did go for these sort of serious chemicals, and we did test them, and they worked quite good. But you'd never get them in here, so what was the point? But the point is that we were able to at least screen some of these to understand what worked and what didn't work. But here's a cool thing, because we were talking to normal practitioners, and they were kind of guiding us to like what plant bioactives might be useful. And through another collaboration, we found that kānuka, there was new kinds of molecules in kānuka that if we showed it, well, I say showed it, but we've exposed it to Phytophthora agathidicida, it actually helped diminish the growth. And so that's something that we've been exploring along those lines too. But the big problem is yeah, you can find all these bioactives, but how on earth do you spread it out in this forest that it actually makes a difference? And that's part of the problem. And it's near impossible, because you can't actually, once something's in the soil system, you can't actually get rid of it in the native forests. So that's kind of, we are still pursuing those lines. There's opportunities there, but we kind of need money, because here's, the government announces we got this many million for biodiscovery, and it's 42 million and it's 6 million a year. And I know that sounds like a lot of money, but here's the facts. If you want to make a drug, you need $2 billion and you need 20 years. And no one's ever going to pay for that in our natural environment. So unless maybe Bill Gates or someone's feeling generous that they might want to do this, it's kind of not going to happen. So it leads me on to the next part of the story, is about how can I understand what makes a system resilient, because that's actually what I'm interested in. Our systems, our forests get a lot of disturbances. We have logging, we have people, we have weeds coming in, we have all these diseases coming in. So what actually makes them resilient, and how much more can they take? Or if they had enough. And so part of what I'm looking at is the link between seabirds and our forests. Because once New Zealand, and I'll go on to this next page which will show you this cool map that was done by a PhD student in Auckland, but once New Zealand was covered in seabirds. Okay, but they all unfortunately have disappeared from the mainland, mostly due to habitat destruction and pests, your ferrets, your stoats, your rats, because a lot of our seabirds are weirdly ground nesting, so they just can't escape and they sit there and their chicks sit there for 3 months. But also we've had land use changes, we've had clearings, we've had cattle and pigs come in and that, and we've had biosecurity incursions. Now there's been a lot of argument about when this came in and kind of proving that it was here naturally and, Captain Cook brought it in or Māori brought it in or something like that. But there's kind of a pointless argument because there's nothing, it won't help us in terms of trying to do something about it. So it's not an argument we tend to pursue. But a lot of people have pursued it in terms of looking at molecular clocks. And of course they've used models that were very inaccurate, and so that tends to give you a gap between either somewhere between 50 years or 500 years. It's again, in New Zealand's history of people, not particularly helpful. So this is the cool map that I was talking about, and this kind of leads into some of the hypotheses I thought, talking to kaumātua, just how thick our birds were in New Zealand, and they were everywhere, like literally, and this is, so Andre's done a screen of New Zealand, it goes, the blue, the purple bits kind of telling you the sort of spread of things. So the blue bits is based on fossil data and some other sort of isotope data. As you can see, the blue bits are different concentrations and taxonomic data. You can see it covered much more extensively than it did today. And then you've got the purple where it's not. And so that's what we're looking at the moment. And it's kind of why I study Aotea, which is in the Hauraki Gulf, because it still has existing bird colonies in it. It's the black petrel I'm particularly interested in. But why birds? Like, why am I interested in bird and what's the link there? And it's because the only natural source of phosphorus in New Zealand soils is from seabirds. It's marine input. And so if you take out, and if you know anything about phosphorus, it's what you need for an ecosystem to grow, right? So when you farm, you pile on nitrogen because that's what you need to start a system, and then you add phosphorus, and that's what our fertilisers are. So if you take phosphorus out, you aren't likely going to have a system that can sustain itself for much longer. It becomes phosphorus depleted, and then you don't get any recruitment and so on and so forth. So it's a very complicated cycle, but essentially phosphorus is needed for forest growth and health. And so what happens when you take out the seabirds, who would have been in their thousands, the biomass would have been huge, you have no phosphorus input. But what does that mean in terms of soil resilience? And there's the kind of work that I'm interested in right now. And partly it talks to conservation, and it's if we do find a link, and I'm not trying to funnel my results into this kind of conclusion, but it kind of stands that we should put a little bit more effort into pest management so that when we can recruit the birds back into their nests so they can help maintain the forest at the current state it's at anyway. So my real passion is soils, and I'm interested in soils and the soil ecosystem and how soils and plants interact, and that's partly where my interests lie in this forest system, because we know that plants create the ecosystem below the soils that help everything function. And what does that mean when you get things like Phytophthora agathidicida, which comes in the soil? And, if you want to know the theory, at the moment it's when the forestry system started up post-World War II, they started to bring in different kauri species from around the Pacific, they suspect it came from there. And that's partly what it is. And we can kind of see that when we look at nursery records and we look at places like Aotea and Waipo where they started up new nursery systems, that it kind of fits with what we see visually in the trees as well. But I'm interested in the soil, and another kind of cool fact is that the soil holds, if you had a handful of soil, it holds more diversity in your hand than you would above ground. There's millions and millions of species of fungi and bacteria and protozoa and viruses and all these kind of microscopic things that I'm interested in. But I'm not really interested in like who's there, but I'm kind of interested in how they function as ecosystem, because everything we do has to have a biological function to it. So when we do put on nitrogen, that all is cycled biologically, and that's where the plants get it from. And so that's what I'm interested in, really. And that's a measure of disturbance for me. It's like we have all these impacts, we take away trees, we plant different trees, we have pasture that's going to change it. we have fertiliser input, or we don't have bird input, and we also have all these microorganisms where they shouldn't be coming in. And what does that do to change the function? And when I say function, I mean its ability to store carbon, right? Its ability for us to grow things, and that's what I'm interested in. So that's partly why I study what I study. I don't have the funds to develop tools, but I can understand how we can make our ecosystems more resilient by bringing back certain components that will help that. And this is the most complicated graph I've got. So, but there is other studies that have looked at this across the Pacific. They very much care about this in the islands because, again, seabirds are the only thing that bring in these kind of nutrients. We don't have fertilisers, so this is what happens. And this is one of the ways they look at it. They look at 15N, so that's a biological signature of influence. And it's been shown that that has influenced the other biodiversity. So once you've got birds and you've got that signal, it has shown that plant diversity is different. I wouldn't say it increases and decreases, that's a question for another time, but they've also got other things in the soil that is present when they're there than they're not. And these all help to function as one ecosystem. And typically the more biodiversity you have, the greater biodiversity you have, the more resilient a system is. And so that's what I'm looking at. And this is just a sort of side diagram of how we do this. We gather soil, we extract the DNA, we send it over to Singapore, usually because it's the cheapest, and it comes back as piles of information. We then apply bioinformatics and NESE, which is our supercomputer, to try and understand and piece together these DNA fragments so we can see who's there, and of that make sense of who's doing what, and does this help with resilience. And just a pretty picture, because I love this. This is up at Aotea, and these are one of the birds, the black petrel, Taku Kitai is their name, it's known, and they're so weird because people, when I talk overseas, it's like they burrow in the ground, like they're weird, they burrow in the ground, and then you see them and you think, that's just some hole in a tree root. And it's actually a bird burrow because you stick your hand and they bite real hard. And so, I had the pleasure of going in and checking them with Biz Bauer, who's an amazing conservationist who works up there monitoring the 3,000 pairs of birds that come every year and make their homes in these forests at the top of Hiraki Mata, which is the big range around there. And so, and that's kind of what keeps me coming back to science, that I feel like I can do something good. And these things are so dang cute. Like, they're really cute, and I do love them, and they're quite special. Yeah, so the helicopter picture is there for a reason. So I was so excited that after seeing my birds and going back, my last sampling round, which was just for Easter, I broke my leg. And I should know better. And the worst thing is when you get older, like you're in your 40s, and you're trying to keep up with your 20-year-old students, you forget that you're not fit anymore. And, you were trying to do something, and I just had surgery, and I broke my leg. So I knew that was happening. And of course, I didn't bring my PLB. I left that on my desk. I didn't bring my first aid kit, and I ran out of battery on my phone. So these are some takeaway points. When you do this, take all that. Don't leave it at home like me. I had lots of my colleagues poking fun at that, but they also thought the helicopter ride was awesome, and it was awesome. I've got to say, it is awesome. I'm so grateful for Westpac Helicopter. But yeah, so that's the takeaway point. But I had sour worms to keep me going until they picked me up, which is only 40 minutes, and they're great too. But take a phone charger, actually. Take a phone charger if you get caught in that, because you can't take anything on you. And there's no phone charger in the hospital, so that's great. But also, my postdoc and my students who are with me, they kept telling everyone that she's been picked up by 4 men in a uniform. And I don't think I'll ever live that down, so I just bring it out in case someone else does. So I guess the more sciencey takeaway points from this is understanding what makes ecosystems resilient is complicated, and it takes time. And there are parts of the ecosystem that will recover, sure, but they're never going to look the same again. And I know that looking at some of the ecosystems down there and other parts where I've worked as ex-geologists in the mining areas, there are places that will never recover. You'll never grow anything down there again. And that's what that is. That's kind of why it kind of scares me, listening to some of the talk that goes around about, let's just mine it and do this. I have seen what happens, and I know that you can't replace, once it's gone, it won't grow. But also, unlike agriculture, which is another area I kind of work in, there is no money to do conservation. And so conservation very much depends on partnerships, and that's what's so key. Coming to Nerd Nite, but probably talking to my own people, but getting the word out there so that people will understand that it's actually the partnerships and the community and we call it whakawhanaungatanga, but the camaraderie that helps make conservation work, because without it, there is certainly no money, but this is what keeps it going, and that kind of what keeps me going is that I can actually do some good out there and working with communities. I haven't had a card, so that's great. She's about to give me a card. But I'd like to thank Nerd Nite, Nicole, for helping me, asking me. I know it took a few months to get me here because I was travelling a lot, but thank you so much, and thank you, the audience, for listening to me rabbit on at this night. Thanks.
Thank you so much, Amanda. Very informative presentation. Are there any questions in the audience?
What a surprise.
I know.
Hey, Amanda, how you doing? My
okay.
question is about the test you maybe finished developing, or is that still underway? Tell us a bit more about that, because that sounded really cool.
It is cool, and it's kind of game-changing, because even though we've developed it specifically for that Phytophthora, it can be used anywhere. And the idea is to make genomics, what would you call it, accessible to non-scientists and community people, so that they're not reliant on the bottleneck of labs and scientists to actually assess the tests. It's kind of like what happened with COVID. So you know how everyone rushed out, swabbed everything and stuck it in a PCR? Well, this is the spit on the version. Yeah, we'd probably be about a year away. Yeah. Yeah, no, I actually don't know, because the pipeline to develop something like that, you know, in a commercial sense, is quite far away. And you'd be hoping it would get down to under $20, put it that way, not $200, $400 actually where it sits at. Yeah.